like to bring this meeting of the Entomological Society of Washington to order. Gary Hevel, would you please read the minutes from the last regular meeting?
Yeah, I'll be reading two sets of minutes, also for the annual banquet. The first one is the regular meeting. The 137th regular meeting of the Entomological Society of Washington met at 7 p.m. on the 4th of May, 2023, and the WG33 of the National Museum of Natural History, and was called to order by President Matt Buffington. 14 members and 3 guests were present, with 22 virtual attendees. Recording Secretary Gary F. Hevel read the minutes of the April meeting, and these were approved. President-elect Don Weber appeared virtually to announce that the annual banquet will feature Dave Wagner from the University of Connecticut and is scheduled for June the 15th. The event will be at Marymount University in Boston, Virginia, and attendees are asked to bring food for a potluck. Past President Lourdes Chamorro noted that she was continuing work on the ESW website and will soon add members to the site. Robert Kua introduced the speaker of the evening, Postdoctoral Fellow, Dr. Giovanna M. Dassel Martinez, whose presentation was the relevance of natural history collection in the genomics era, the parasitoid blocks of the family break Conidae as an example. Dr. Dassel Martinez began by noting that there are over 21,000 species in the subfamily Draconinae. The family is divided into 41 subfamilies and now includes Gasteroptidae. In her genomic studies of specimens, she found serviceable DNA in 90-year-old museum specimens. The Smithsonian collection includes some 250,000 dried specimens with 2,600 primary types. Museums are especially valuable for finding extinct species for scientific studies. Assessing diversification rates in Draconidae provides better general knowledge of the family. The meeting was adjourned at 8:05. Now, skip right into the annual banquet, and then we can criticize the minutes at the end of this. The 2023 annual banquet of the Entomological Society met on June the 15th. President-elect Don Weber secured a venue at Marymount University in Boston, Virginia, and attendees were quite favorably impressed with the facility. Members and guests brought potluck dishes to the event. Approximately 50 members and guests were present, with a guest of 30 virtual attendees in attendance. After the meal, President Matt Buffington and President-elect Don Weber introduced the speaker of the evening, David Wagner from the University of Connecticut. The title of the presentation was Using Caterpillars in Biosystematic and Ecological Studies and as Beacons of Climate Change Befallen the American West. Dr. Wagner is currently working on a multi-volume guide to for the caterpillars of Western North America, which will be published by Princeton University Press. He showed images from his work, then discussed advantages and disadvantages of using larval data in the systematics of Lepidoptera. Climate change is easily recognized in the Western states and will be devastating to wildlife in the future. President-elect Don Weber deserves applause for finding a great venue and a leading scientist of entomology.
Thank you, Secretary Hevel. Do we have any or corrections from the last regular meeting? Brendan.
Gasteroptidae are not Ichneumonidae.
Have they been thought of as recommended in the past?
No.
Okay.
Yep.
Giovanna's done some pretty amazing phylogenetic work, but I don't think she's yet figured out how to get Gasteroptidae into the Bracanas, so.
I'm pretty sure. Yeah.
Yeah.
Are there any additions or corrections to the minutes from the banquet?
Yes.
One of the grand things about the evening was that we got to take the subway all the way to within 3 blocks of the restaurant.
Yeah.
Some of us walked a block to with no subway. Oh,
Nice.
we live in Boston.
Oh,
Oh, you're lucky. Yeah, yeah. We love it when you guys find Boston for things.
I might say one more time, the sign-in sheet has been circled.
Here it is.
Circled 1 or 2 people up front.
Okay, I'll pass it back. Let's put it on
Can I get a motion to have the minutes approved as read? And
So
with the
moved.
amendments? Second? So moved. Excellent, thank you. Okay, officer reports. How about President-elect? Would you like to say a few words, President-elect?
Right now, Matt, since you called on me, we are in the process of forming a nominating committee to fill the offices of curator, which we have a very good prospect, editor of Proceedings of ESW, which is TBD, and president-elect. So if you have interest, and I hope you do, in serving on the nominating committee, see me. You cannot be an officer, is that correct? I think that's correct.
I think that's correct.
And even more so, if you have interest in any of those open offices, please see me.
Yes, just to make sure everyone in the room got that message and everyone there in TV land got that message. We're looking for some officer positions. The two critical ones at the moment that we don't have names for: editor of the proceedings, which I would suggest we might move to a co-editorship because of the labor involved, and president-elect. That's the person that's going to become president. So we have a good idea of someone who might be interested in curator. So that position is kind of already, but the competition is the American way, right? So
Yeah,
we
it's
might
an
have
open,
a fierce
open,
race.
let 1,000 flowers bloom.
Yeah, yeah.
Chairman Mao once said.
Since you have the floor at the moment, would you like to say something about the tour that you and I are kind of?
Oh, yes.
It's not really an ESW
So,
thing, but it kind of is.
Matt, you and I have to work on this. So you all know, I think, that the Entomological Society of America meeting is in early November down the river at National Harbor, so-called. And on Thursday, immediately following that meeting, Matt and I are giving a tour, and Floyd's actually giving 3 tours, is that correct? Of the Smithsonian, which are already booked. Matt and I are giving one called C.B. Riley's Washington, I believe it's called. So our founder, C.B. Riley, has a lot of history left behind here, including many collected specimens, types, etc. And that will be our first stop on the tour, right, in this building. And then we'll go to his first site of his first house, which he built in Washington, which is still standing, and we're trying to figure out how we might gain access to that. But whether or not we do, we will stop there. It's up near Thomas Circle. And then we will visit the site of, or near the site of his mansion in Calorama on Wyoming Avenue. It might be an interesting detective story to figure out exactly where that is. It's wood, it's gone, it probably burnt down or got torn down. But in those days, Wyoming Avenue was the outer reaches of DC. It was not part of L'Enfant's plan. It was further out. And when you wrote Professor C.V. Riley, Wyoming Avenue town, it went there. So it didn't have even a number. We haven't been able to find that. So that's one interesting thing in that area. The other interesting thing is the site of demise of that C.V. Riley, who you all may know, was riding on a bike with his son and hit a cobblestone and went over the handlebars and had and never woke up. And his early death was quite tragic. Anyway, we have the site of that, and it's listed by the DC government still as a dangerous site for bicycles. It's along, that's Connecticut, and I don't know where that is exactly. Anyway, the foot of a large hill, as you might imagine. And then our last, our final stop will be the National Agricultural Library, which of course C.V. Riley's papers, his desk, various other memorabilia are kept, and we will have lunch there. And as part of signing up for that, if you want to, you get a free book. This biography of C.V. Riley, and I will sign it for you. Matt will sign for you if you want. I don't care.
I'll sign someone else's name.
Yeah, whatever. Yeah.
So we still have spots open.
Yeah, so we still have a few spots open. I think maybe it didn't get the promotion.
She said it's the Thursday after National Harbor.
Yes,
November 9th?
it's the day, yes,
The 9th.
it's the day after the meeting, and we did that by design. We just did not want to pull people out of the meeting.
I think that's probably part of it is.
Yes.
If you've ever seen the
People are-
ESA meeting, you know,
Yep.
Thursday morning, it's like lights, cockroaches when the lights come on. Everybody's like,
Yeah.
Yeah,
So-
maybe you're right. Okay, well, there's still some open spots, and what does it cost? $75, which is like not much more than the cost of the book, so.
Lunch and a book.
Yeah, there you go.
Yeah.
So anyway, for what it's worth.
How long is the tour?
What did we figure? Well, we end up with lunch, so, and when does it start?
I
9?
can't remember.
So I think it's more or less 4, no, it's 4. You better keep the schedule. It's 4 to 4.5 hours, and then we gotta come back. So it's probably figure not, I figured 9 to 2.
Walk around.
more or less. Yeah, we wanna walk around. Yeah. And if it goes well, we may have a ESW reprise at some point,
Oh, yeah,
especially if we get into that building.
Yeah. So it is the site of the first meeting of this society, this house.
Oh, that's true.
That's right.
Yeah. Oh, yeah.
Where did you say we should find information about it for?
Okay, so if you go to the Entomological Society of America, ensoc.org meetings, it's kind of buried in there.
It's like field trips. There's like,
Field trips.
there's a schedule by day, and each day there's like, so you'll see Floyd's tours on there.
It's field
And you'll
trips.
see ours.
Okay, if you don't mind, send our email to this.
That, no problem. You can
That's a
do
good
that.
idea.
Yeah.
Oh, we can put it on the website.
Put it on the
Figure that
website.
out, yeah. Yeah.
And if anyone has any bright ideas of folks that are going to the meeting, either the Entomological Collections Network meeting and they want to promote the society, definitely, we haven't done any formal, but I would love to kind of use it as a springboard for making sure people there know that we're still like around and we're not just some local club. Yeah. All right, thank you, President-elect. Treasurer, not here. Program Chair, would you like to say a few words?
We have 21 Zoom participants.
I think you, oh, sorry.
Lined up our next.
Jesus. Yeah, so we're celebrating our postdocs here in the fall tonight, and then the next meeting is Brendan Budno from Hymenoptera, who actually is here tonight. So they'll be next speaker. And we have a December speaker as well, right?
Dave.
Oh, excellent. Our very own. Cool. Okay. I think that's it. Curator, we don't have a curator at the moment. So we'll stay on that one. Okay, at this point, I'd like to move on to the introduction of new members and visitors. Oh, we don't have a membership secretary report. That's it. Elizabeth Young, she's not here, right? That's- Would new members that are here tonight, or visitors here tonight for the first time, like to introduce themselves.
Hi, I'm Lisa. I'm a high school student. I was at the banquet in the spring, but I'm here for the first time. I'm happy to be here.
Cool, welcome.
Thanks.
I'm Pedro. I'm from Brazil, and I'm making an internship for 6 months with Hannah in the arachnology. Nice to be here. Nice to meet you, everybody.
Nice to meet you. Oh, you go. I'll go next. I am Alexandre. I am a grad fellow in New York, and I'm a PhD student working on different flights at Biala and Thorsten. Nice to see you.
Hello, my name is Jeff. I'm a first year grad student at George Washington, and it's my first meeting. So yeah, thanks for letting me be a part of it.
And I'm Jill. I'm here as a postdoc for 6 months. I'm working in Hymenoptera on ants. And it's nice to meet you all.
Is there any unfinished business from the last regular meeting that we can wrap up at the moment? Is there any new business people would like to bring to the floor at this moment?
Is this including exhibits?
Not yet.
Not yet.
Hold your horses.
Okay.
We're getting there. The only horsing around.
Yeah.
I would like to introduce one piece of new business, and that is we're hoping to get the attorney's comments on our bylaws so we can actually formally incorporate, and then that means we can start formally investing the funds. So those comments are with Abby Kula, the treasurer, and she hasn't moved them to us yet for the rest of the executive. But once we're through them, then we will make whatever changes are necessary and then bring them to the membership to be approved and voted in. So keep looking forward to that one, because I know everyone's super excited. Okay, now is a presentation of notes and exhibits. My guess is Dr. Davis has something.
Yeah, I got a book here,
All right.
and this just came out. It's a beautifully illustrated book. And David Grimaldi is a primary editor, and there are 5 chapters in this book. Dave Grimaldi was wrote one of the chapters, and Michael Engel wrote one of the chapters, and my son Stephen Davis wrote one of the chapters, and I don't remember who wrote the other 2. But anyway, it's a beautifully illustrated book. You know, it's just tons of photographs, and it's called The Complete Insect. It says it's on anatomy, physiology, evolution, and ecology. So those are the main chapters.
Can you pass it around? Is that okay to pass it around?
Sure, it's heavy.
Make sure you don't have a
Don't
piece
drop
of
it.
grease on your hands.
Yeah,
That's right.
Any other exhibits? Any other? Oh,
I would just note that in the Daily today, not be confused with the Daily Show, the New York Times podcast.
Yeah.
They were talking about mosquitoes. How the mosquitoes are winning. Despite all the money that's allowed in the fight there.
Was the emphasis on resistance or what? What was that?
Well, there's a number of reasons. There's an invasive mosquito in Africa now that came from Southeast Asia, and it's likes urban areas. So malaria had largely been a rural problem. Urban areas, and there's resistance to the insecticides that were developed. They didn't mention anything about like some of the new ideas for genetic engineering.
It was also recently in the news that they're quote unquote fumigating Paris for mosquitoes. And also they're worried about bed bugs, especially the Olympics.
Another thing they mentioned was like climate change, changing the areas where certain disease areas.
Don't we have, wasn't there something in the news that there's now the malaria carrying mosquitoes in Maryland?
Yeah.
One instance.
confirmed case of. the-
Yeah, one confirmed case.
They're calling it malaria list.
One more?
Did you get that, Gary? Did you get that?
Yeah, we can do that. It's so memorable.
Alan's sharing something on his computer.
It's reclaim the host, or whatever that means. There are 4 people in the
Well, I
waiting
know.
room apparently too.
Oh.
Oh, yeah, that's Al. Oh,
Alan's in charge of that.
but maybe try it.
Reclaim host.
No, just.
Oh, it's gone.
Yeah.
You got an announcement?
Okay. While we're doing that, there's another exhibit over here.
Yes,
Yeah, go ahead.
yeah, I ran into Matt yesterday on the metro. And part of my thesis was starting to draw bugs, create my own forest, super broad term. But yeah, he immediately recognized it and told me to bring it along. So I worked on it a little bit more from what he saw yesterday, built the frame and.
Yeah, it's like 3 more species on there than just
Yeah.
yesterday. So that's
Yeah.
pretty fast.
So that's it.
Yeah,
Thanks, man.
Oh, thanks for coming. He's looking for inspiration. I thought this would be a cool group of people, with lots of different backgrounds and such, so.
Like, I found it, so it's on the screen here, and I hope it's on.
Insect.
Bob Copeland, our friend and colleague in Kenya, started doing this thing, he calls it Insect of the Week. So he's up to number 41 there. Every week he has a little story about a particular insect. And he put one nice pictures.
Is there a way to
Those.
subscribe to get the emails?
Well, yeah, I think the only way is to contact Bob.
Okay.
This week was printed. He was asking me, is this the largest printed in the world? I'm not sure, it's pretty big. It's 2x. Anyway, kind of reminds me of, I don't know if anybody, this Quint Wheeler podcast.
They're
Species.
It's really great. Yeah, I
Yeah.
started listening.
They're like 5-minute things. It's kind of the same thing, a couple paragraphs. Usually something interesting about the species. and talks about the-
Yeah.
What's the name of the podcast?
So Quentin Wheeler has a podcast called Species, right?
Yeah.
Thank you.
It's kind of interesting because he goes on in this kind of monotone voice.
Yeah.
What he's saying is like really.
Yeah, yeah.
That-
It's a good way to pass time.
Yeah, we should ask Bob if he'd be open to us, like,
Posting it on our website.
yeah, something like that. I think he'd get a kick out of it.
I think it's an archive.
He definitely keeps an archive, but they're not posted.
Right.
Make like a circle group.
Yeah, Brendan?
Just thinking about insect news and the importance of museums is potential RIFA in Italy, right?
Yes.
What?
Yes, so Sicily.
Explain.
So red imported
Oh,
fire
yeah.
ant has been
Oh, God,
detected for the
Rifa.
first time in Sicily, which is, that's big news.
I'm sure they've got a real organized plan.
What happened? Will you take the day?
Red imported fire ant in.
Something like 19 colonies that's been discovered.
Oh.
Probably a lot more.
Really bad.
Really bad. Yeah.
While
Pretty
we're
good insect news.
While we're on Europe, I do have some, well, I thought it was good news. I went to Aveiro, Portugal, for the IOBC, International Control Western Palearctic Regional section. And this was on the benefits and risk of biocontrol agents. And it was very interesting because there were weed biocontrol people and arthropod biocontrol people, and they don't often get together. But it struck me how many interests in common we had, that in particular, our interests tend to focus on herbivores, either suppressing or encouraging them. And a local example there was the long-leafed acacia, which is being controlled by a galling wasp. And there it was out on the dunes with galls and the bushes dying. It was very, very dramatic, as were the flamingos, the local bird fauna. And the eucalyptus, we had a tour relating to eucalyptus. Now, if you can imagine, parts of Europe have a love-hate relationship with eucalyptus. And some of them love it, and some of them hate it. And some of them say it's invasive, and others say, no, it's not. Great. So that was interesting to hear all those perspectives. But that was, as far as I could say, good news. There were some other really good news. One was they have introduced, or actually one of them showed up, a parasitoid of the citruscillid before the citrus greening pathogen has shown up, so that they can suppress the citruscillid. And this is in northern Portugal and Spain, and it hasn't gotten yet into the citrus growing areas of Portugal and Spain. They may have a real possibility to suppress the vector enough so that the citrus greening won't get established. So that's some good news.
That's some good.
If Sicily can get their act together. I'd like to mention though, International Organization Biocontrol, what we could say sister small entomologically related organization, has a symposium and membership session at the Entomological Society of America. And there are often some really interesting presentations by the award winners, which include a master's, a PhD, and a junior and a senior researcher. So you're invited, and we're invited to join to the fun.
Yeah, especially for all you visitors, this is the most recent hard copy journal from our organization, October 2022. And that's probably because of the shutdown from COVID,
There's
right?
a number of combination of factors.
Yeah, lots of things like that. This is from the library, our entomology library. And several well-known authors are listed on the cover, including John Burns with 2 articles. He's the most senior of scientists in our Department of Entomology. So I'll pass this around as well.
Did you bring that art and stuff you got from?
I was handed our aphid systemicist Gary Miller retired last Friday, and he gave our current RL Night Gates a massive wad of course in stock of Washington correspondence from like the 1950s. And it's just, a lot of it's pretty basic, but it's so fundamental to see like how the organization was run, same issues, same, just instead of emails, it's triplicate, carbon copy. So I wanted to bring it to exhibit, but I need to kind of go through it first, make sure there's any PII in there. I need to suppress that.
Okay, that'll work for one meeting cycle.
Yeah, exactly. Yeah.
Were they trying to get us incorporated?
I really, it'd be fun to go through and kind of make a summary, like what are we still trying to do? Cool. Okay, I think we are ready to move on to the presentation of the evening. Program chair, if you'd like to take over, please.
Okay, it's my pleasure to introduce tonight's speaker is Allan Cabrero. Allan received his PhD from Berkeley earlier this year, where he was co-advised by Kipling Will and Patrick O'Grady. For his dissertation, Allan studied the phylogenomics of bee flies, which we'll be discussing tonight. He's currently an NSF postdoc at the Smithsonian Department of Entomology, working with Torsten Vico on genomics of Basiloidea. And this is actually Allan's second stint at at the Smithsonian. He was an NHRE intern here 9 years ago. I guess that's in the summer, right? So, oh, he get to come to a ESW.
I came to the banquet. You guys had to.
Then I can say welcome back. That's fine. Yeah.
Everyone, thanks for having me. Can everyone hear me all right? Well, it's great to be back almost 10 years from now. Yeah, today I'm going to talk about bee flies and kind of what I did during my dissertation at Berkeley. You know, there's a wide range of topics from microbiomes and then heat shock proteins and phylogenomics and taxonomy and kind of what fueled my interest. I mean, we're all obsessed with our insects that we study, but I really enjoy learning about all aspects of bee flies. So I kind of want to learn as much as I can about them, about their evolution, diversification, their taxonomy. And generally, I think they're one of the cutest insects on the planet. So if you learn anything from my talk, I hope you'll accept them as the cutest insect you was. So today I'm going to talk about what are bee flies. It's like the common photo you see floating around the internet. You know, these very fuzzy, cuddly things. They kind of look like flying cotton balls. They're very diverse. Not all of them are fluffy. Some of them are, you know, not as hairy, but they are colorful, and some have this, you know, weird humpback appearance. And I would say they've kind of made it big in the insect world because they are famous. This is a Pokemon called Cutie Fly, and it's based off of a bee fly, specifically this genus that I studied for my dissertation, Ordotus. I think that they're also really interesting, and we've all heard about, you know, there's been some debate with, you know, bee flies being described in photos. So I would say they're a really charismatic group that most people are familiar with. And so where are bee flies found? They do really well in arid and semi-arid habitats, so Mediterranean climate or wine country. You can find them in Southern Africa, the American Southwest, you know, the Mediterranean, Australia, Chile. And they just do really well in desert environments. And there's about 5,000 described species. I have some photos of their habitats where I visited. So this was the Allan Cabrero super bloom in 2019. It was just an incredible diversity of bee flies. In some canyons, I found up to 30 morphospecies of bombolillas. So it's pretty astounding how much stuff was found. I've also collected them in South Africa. This is the succulent Karoo habitat, which is the most diverse region in the world for succulents and flowering plants. And typically with bombolillas, you find them on really hot, sunny days. So when you're out at noon, when most things are calming down, bee flies are the most active. And so that can make for interesting field work, right? Because you're really waiting for the heat to come up, and then while everyone wants to take their siesta, you have to kind of trudge along in the heat. But even on cloudy days, so this particular day, you know, if you're done collecting in the desert, you know, windy and cloudy days are really not the most fun to do collecting. And even on this gloomy day, I found about 15 morphospecies of bombolillas. So the diversity in Southern Africa is incredible. I've been to Namibia, so this is shot through my iPhone. It's one of my favorite photos. And I've even found bombolillas on the top of the dunes just flying by. I got kind of lucky and I noticed something hovering by and I was able to catch it. Find them along the beach. I kind of ignored the water. And on these daisies, I collected this really fluffy bombolilla. So really found them in a variety of different habitats, but most of my research has collected them in arid regions. I haven't really tried looking for them in a rainforest or tropical environments. So a little bit of their natural history. Adults are nectar and pollen feeders, and they're very important pollinators of desert flowers. Some studies in South Africa have found them to be the most important pollinator in the Cape Floresic province, especially for these type of daisies. And you can see these petals that the flowers produce that are modified to look like a bee fly. And so this genus Megapalpus, the males will aggressively mate with the flower petal, thinking it's female and pollinating these daisies. And so researchers in Southern Africa found these to be more important pollinators than bees and butterflies. So, but no one's really looked at the pollinators in like the Southwest or Australia, but given the large amounts that you find them during the flowering season, they're probably just as important. One of the fascinating things about them, so they're kind of cute but also deadly, is they are parasitoids. And so for those that don't know, parasitoids develop by drawing the nourishment from the host. There is bombolilla records for ecto and endoparasitoids and even some hyperparasitoids. So here's a cool photo of a tiger bee fly larva. And you can kind of see the bumblebee and maggots here. They're kind of cute, right? Maybe not cute for a bee fly. So the first section I'm going to talk about is the role of host phylogeny in shaping the diversity of bee fly microbiomes. So I'm going to talk a little bit about what a microbiome is. So if you imagine your host genome, and your whole of genome is this concept of your genome plus your microbiome. So your microbiome is the genetic content of the host microbiome communities. So this includes bacteria, archaea, viruses, protists, and fungi. And for the bee fly, that's what we care about. So there's two types of microbiomes. There's your transient microbiome. So this is influenced by your diets and your environment. So depending what you eat can shift your bacterial content when you travel. So, you know, like traveler's gut or whatever multitudes are banned whenever you go to other countries and you feel sick, that's probably a shift in your microbiome. And there's your resident microbiome. This is just kind of stuff you carry with you throughout your life. And things are just a constant in your microbial community. And so what really got me interested in microbiomes is reading these papers of how much microbiome can influence your, you know, acquisition of research, nutrients, your speciation, your behavior. There's all these crazy studies of like just off of the host switching with the change of microbiome. There's a lot of studies in parasitoid wasps like Smithsonian. And so I'm going to talk a little bit about some of the host microbe interactions that have been found in insects. So here's a parasitoid wasp. Studies have shown that microbes can act like a secondary immune system, so they can protect larvae from parasitoid wasps. So the bacteria will attack the egg that's laid inside, you know, the wood-borne larva or something like that. They can affect development and reproduction. Studies have shown that if you remove the microbiome of an insect as it's developing from a larva, it develops deformed or failures to molt to adulthood. So the microbiome is obviously very important in how, you know, insect larvae develop. And I remember years ago going to a cool talk where they studied crustacean larvae in the ocean, and removing the microbiome, the crustaceans never molted to adulthood, they just stayed larvae forever. So clearly our microbiomes are influencing us, they're influencing insects. We all know about aphids and how they're very tied to the microbiome, and it influences how they, you know, are able to access new food resources. And the coolest stuff I read about, especially in Estonia, is how your bacterial community can influence where you lay your larvae, how you behave. This has even been shown in mammals. So, you know, high bee strains will avoid other packs that are too different from them, and changing their microbiome will cause them to mate preferentially those with a similar microbiome. And that's been shown into frittids as well, where they'll lay eggs on fruit that have like the same yeast or fungi available to them that attracts them. So how does this fit into Diptera? So most species of flies that have been studied in the context of the microbiomes are things that are medically or agriculturally important. So, you know, our mosquitoes, our Drosophila, some frittids, there's some studies on the tsetse fly, but all of them kind of center around Wolbachia or bacterial behavior. So no one's really looked at other aspects of flies, and especially a parasitoid fly. So there's a bunch of studies on parasitoid wasps in Estonia, a lot of cool research there, but in some ways I'd argue Diptera are kind of more interesting and cool parasitoids than wasps, because they've evolved it so many times independently and they do so many weird things. Sorry for all the wasp people, you guys are giving me 30 minutes, but I think overall flies are more interesting than wasps. Sorry. So what I wanted to study, where I was interested in bombolea is, you know, do they have a microbiome? And if they do have a microbiome, you know, what is influencing? Is it ecology? Is it phylogeny? Is it just random? Do they have a resident microbiome, or is it just transient? So my taxon sampling, I collected individuals throughout Southern California. There's about 36 individuals across 8 subfamilies and 18 genera. And we did marker-based sequencing, and this is mostly through 16S RNA amplicon sequencing. So it's one of the standard microbiome sequencing methods. We used the UParse pipeline, and so just kind of a figure of that. And then we did analysis in R. So we got about 400,000 reads, about 1,500 bacterial OTUs, and this comes out to about 115 bacterial OTUs per bee fly host. Here's kind of a species accumulation curve that just shows that given our sampling method and our sequencing, we kind of captured as much of the microbial diversity as we're able to get. This figure, is just kind of showing like the larger OTU families we found across samples. So these families were the most common across all our samples. which I'll explain more about what these families mean. Here's kind of a breakdown of the phylum of different microbes that we found within bee flies. So a lot of proteobacteria, that's the purple. In orange, we have a lot of cyanobacteria. Some of these samples are kind of interesting, and I think that's the sampling bias because we didn't have multiples of each genus. So if we had more sampling per genera, I think these results would have been more balanced and equal. Here's where we kind of did like a PCA plot looking at parasitoid type, so and by subfamily and their habitat, and this isn't really telling us anything about the bacterial community composition, so there's no pattern. It just kind of seems to be all random, but of course this can be biased by sampling as well. These are the results that I got really excited with. So we looked at all the genera of bacteria species found within bombyliids and kind of dug through the literature to find out what do they do. So we did find a lot of Staphylococcus, which is bacteria associated with pollen and nectar feeding. So that makes sense, right? Bombyliids are pollinators, so we expect to find this bacteria. We did find this Actinobacter, which is that bacteria associated with larval development as soil bacteria. So bombyliids get in the soil. It makes sense that they have this bacteria. This is really cool, the mesoplasma in red. This has been found in leaf-cutting ants and predatory army ants, and it's thought to be bacteria that helps with the processing of chitin. And so you might be asking yourself, why does a bombyliid need to go and process chitin? Well, their larvae feed on other insects, they're parasitoids, so it makes sense for them to carry this bacteria. And of course, we found a lot of Enterobacteriaceae. So these are your symbionts. This is where the aphid, symbionts are found. We're not entirely sure what Enterobacteriaceae they have, but they are carrying some symbionts. And I would also say that this table is what was found in 75% of all samples. So those kind of hints at possibly if this is like a resident microbiome. So in microbiology, there's a lot of different species of bacteria that all can do similar things. So it's really hard for a host to carry just one bacteria because there could be 10 others that do the same function. But the fact that we found these across 75% of all samples and a wide geographical range just kind of shows that this might be their core microbiome or what they're constantly carrying around. And so summary and future directions, we have a baseline of like the bombyliid microbiome. It's the first like assessment of a parasitoid fly and looking at what kind of microbe they carry. Moving on, it'd be great to do some more shotgun sequencing, which can really help us understand composition, structure, and the function of bee flies. And long term, it'd be cool to do like lab studies where we rear bombyliids and remove the microbiome to really see what's happening there. And if everyone has a great idea on how to rear a host and then rear a bee fly and keep that system going, let me know, because that's kind of a challenge. Got to rear the bees and then hope that the bee flies visit and somehow keep it. Any questions about this before I move to the next section? Or we can take questions for you.
Just real quick, what's the host specificity of bee flies on their host? Like,
you
have monophagus?
So yeah, there's only about 10% of all bee fly host records known. So for all 5,000 plus species, we only know about 10% of them. There's some host specificity, but there's a lot of generalists, and we honestly just don't know the records at all. So we have no clue. And I've talked to Neil Evans about it and tried to figure it out. And I thought of a project of using genetics so some people do this with Hymenoptera. You can sequence Hymenoptera just as it emerges and see remnants of host DNA to try to get at what bee flies are on. But that was only like a 10% success rate of gathering that DNA. So there's a lot more work to be done.
So related to that, how many, a typical specimen in a museum is not going to have any host association, right?
Yeah.
And do any of them have host association?
I think if you're lucky and you see them, or presumably if you catch one, like you catch a female ovipositing near a bee nest, I'll collect the bee, talk to my friends and be like, what is this? Otherwise, you just don't know, right?
How would you go about removing microbiome?
So there's like different techniques of like sterilizing them essentially on that literature.
I was wondering what your range of sampling of habitats was.
So I mostly in Southern California. I did some chaparral, which is like Mediterranean scrub, and then I went into the desert, so that was like the main divide. But it'd be kind of cool to compare it to, say, maybe some tropical bumblebees, but I haven't had the chance to collect those. Sure. Cool. So the next section was the phylogenomics of the bee fly genus Lordotus. So this is the genus Lordotus, your classic fly. So my advisor Kip was writing the California Field Guide to Insects, the second version, I believe, and he was told by the editor that, or the publisher, that every insect needs a common name. And so I jokingly said fruit flies, and they published it. So this is now this is now the common name for bee flies, which I love. And these are described by Herman Lowe in 1863, and they're known for being quite charismatically funny. So there's about 29 species found throughout Western North America, and they range from Baja, and I've seen some records on iNaturalist, to just about Mexico City, all the way up to Washington, and as far east as like Kansas and Oklahoma. So a huge chunk of the Southwest. But at least 20 to 25 species are all found in Southern California. So it's a huge range for 29 species, and all of them occurring in Southern California. Historically, morphology was used to delimit the species, and there's 3 major species groups that were defined. But this group has always gave authors a lot of trouble. They've used like average antenna length to delimit them. All the male genitalia is very similar, almost the same. Same with the female genitalia. So male genitalia, female genitalia doesn't tell them apart. Coloration is highly dependent on the population where you sample them, so that doesn't really help either. And so they hypothesized that there's 3 species groups: the apicula group, the miscellaneous group, and the gibbous group. And I won't exactly go down the list, but it's based on the costa and the antenna style being present or absent. It's how they used to break up the genus Lordotus. And they found further species complexes within those groups, specifically around apicula and Sauropodes. So my objectives were to generate the first phylogenetic hypothesis for this genus, and then kind of test does morphology hold up these species groups. Again, this is where I collected them. Can't complain. I like to show this slide a little bit. So they're used to the fuzzy yellow ones, but a lot of them are silvery and gray. And so could you find, there's 2 bee flies in this photo. If anyone wants to try to guess where they're at. Okay, I'll move along. Right there.
Oh, sorry.
So when you're collecting these, you just kind of have to walk slowly, and then you'll disturb them and they'll come back. Sometimes on a trail, you'll disturb a male and he enters another's territory, and they just all fight down the trail, and they'll settle down, and then you just have to remember where they're at, and then you start collecting. So here's my sampling. So here I kind of did in museomics. I sampled a lot of museum samples and freshly collected samples. So 28 fresh, 49 pin. I would have loved to have more fresh sampling, but pandemic kind of changed that. So we relied heavily on the museum samples, and the age varied from 1951 to 2018. But I tried to get at least every species represented that's found in Southern California. And I think my final sampling had every species. So we used UCEs, we used a custom Acylodia probe set that we designed with Torsten collaborators. So we didn't use the general Diptera probe set, we designed our own just for Acylodia. And as we all know, studies have shown that you can use museum samples and actually get a good amount of DNA. So this is just kind of the standard pipeline. We used Phylucci, and we generated a tree using IQ tree with a 75% complete matrix. So we successfully extracted DNA from museum samples, about 12 million paired sample. And this came out to about, at the lowest, 4,000 to 300,000 contigs per sample, which averages about around 38,000 contigs. And then from the contigs, we found about 1,200 UC loci of 2,300, which comes out to about 936 UC loci per sample. So this is the phylogeny. We kind of hoped for something like this, all 3 groups broken up evenly, but the world doesn't work out that way. So this was our results. And there's some things I want to point out. There's a lot of strong support for a lot of these nodes. And some things I wanted to talk about were these species complexes. So this is the apicula group, and it's just a huge spread from these species. And these are all, identified as separate. Some of these are paratype. And so clearly in this apicula group, you do have some species that are coming out with strong support on their own, but this complex is just kind of a nightmare, right? Genetically, they're super similar. morphologically you can't tell them apart, what should we do? A lot of species were also not found to be monophyletic. So the most interesting one to me was this Lordortus schlingeri, which was thought to be part of the yellow group, Apicula. And so I sequenced 2 paratypes that are collected at the same place by the same person, and they come out in 2 different completely parts of the tree. So which one is schlingeri? Which one is something else? I don't know. I have to probably look at the type and compare it to what I collected with the paratypes, compare the morphology, and try to what is going on. Same thing with the species Divisus. They come on different parts of the tree. And there's this species that could be Diversus, and it was found in Baja California, but it comes out pretty genetically distinct from other species, so it could be potentially a new species, which would be really cool. So none of the species groups were found to be monophyletic, and it's clear that these species groups need a lot of work. I kind of got rid of the 3 species groups and broke it down into 2, and that kind of works out morphologically as well. But of course, there's always more work to be done. So I would love to sequence more museum samples to try to capture the breadth of the diversity, maybe do some morphometric analyses to see how much morphology is influencing the species limitation, and maybe look at some biogeographic and diversification analyses. I'm especially interested in the diversification rates because this is a pretty large genus for like the desert. You see these papers of like rapid radiation of lizards, and it's like 5 lizards, which is like cool. But this is 29 species, though. That's a big radiation, so looking into the evolution, which is kind of what took me in this group, is how do they evolve and diversified across arid habitat? I think is really fascinating. Any questions about this aim? No.
You're using UCE, right, which conserve areas. You're looking at a really recent, very recent relationship with the species,
right?
Is the method capturing enough of like more variable and rapidly?
You can with UCs. Yeah. I mean, I think UCs can struggle with like population level, sampling, but we're sampling across the genus.
Just using that method or looking at relationships on gender,
right,
family. Just curious how well it's put.
It's worked with other groups, like vertebrate level.
Are they still keyable to species largely, do you think, or do you think totally?
Like the key will take you in the right, there's a key available and it will take you to at least a species group, but a lot of the, it's like if you find out the female, then it's that, but then the female looks all the same. So there's a lot of, can look like this if you're here. And so kind of a lot of things like that. There's a huge variation against like the pilot and like the coloration of the CD. And so there's not really like one, there's some species that are easy to key out, and there's some like in these complexes that are really challenging.
Are there like, do you have to think about like, are there parasitoids that might be mixed in with some of your, I'm just thinking about why your one species wasn't monophyletic.
Oh, like there were like a parasitoid inside of it? No, I didn't. When I dissected them out, I didn't see that. So I guess I sampled the museum specimens. I took them off the pin and kind of cracked open the abdomen and the thorax so things could seep in. I didn't find it.
So your tree looks frighteningly similar to a tree that I'm battling right now. And I'm just curious, no, I don't mean to put you on the spot, but what species concept would you kind of triangulate given your data? Like, where do you draw the line?
That's a good question. I haven't really decided that yet, just because we're gonna do some more sequencing and then maybe try to figure out how I want to like revise the group and taxonomy. And ideally, I'd like to take like an integrative approach and try to do the morphometric analyses and maybe see there's some methods that people have used with like harvest when they're really conserved morphologically and use morphometrics to really tease apart these subtle differences that we might not notice. It's a lot of measuring, but it'd be kind of cool to figure out like what's going on with this genus like this, because it's stumped many authors throughout its history. Maybe I have to synonymize this complex and just say it's a highly variable species found. Maybe that's what we have to do. I don't know.
Have you tried to extract microbiomes from UCB's data?
I thought about doing that as well. That's like the next step.
Probably, I don't know, like microbiomes are rational.
Yeah.
Okay.
Yeah, at Berkeley, their cluster took me like 2 months to run the analyses, and it ran in a day here at Smithsonian. So now I have faster sequence or technology to play around with. We can do them at the end as well. I have one more section. Okay, so then the last section is kind of what I'm doing here. And it's kind of looking at desert adaptations of asteroid flies, again, genomics, their heat shock proteins, and hopefully climate change. So kind of branching out beyond bombyliids and kind of building on my fascination with, desert radiations and desert adaptations. I think historically we're taught that diversity increases as we head to the tropics and deserts are kind of barren and not diverse, but some groups like Asteloidea do exceptionally well in desert habitats. So Asteloidea is this large superfamily in Diptera. It's about 15,000 species. We have your charismatic robber flies or assassin flies, your bombyliids, your apiosauриds, your mitids, and as I mentioned, they're very diverse in arid habitats. And so I'm going to tell a little bit about like what my thought process was with this project. First, kind of talking about high throughput sequencing. So we all know that technology has increased. We know about comparative transcriptomics, RAD sequencing, UCE. These are all popular methods right now. But a risk of them is to have like a limited usability, right? Like once you get UCEs, there's not really much you can do with that data besides what you got your UCEs for. And of course, the next step is genome sequencing, which can be very expensive. And so we're hoping to get do some genome skimming or low coverage genomes. They're relatively affordable and they're very versatile. You can pull UCEs from them. You can do some genotyping, trait association. There's a lot you can do with a low coverage genome. And so the idea here is to build like a repository or like a foundational sequencing, to sequence a bunch of low coverage genomes across all of Asteloidea. And that can serve as a data set that future researchers can play around with and do a lot of things. So what are heat shock proteins? So they're ubiquitous conserved proteins. All of life has them. They protect cells during stress, typically expressed during high amounts of stress. So it's commonly associated with intense heat. Heat shock proteins protect your DNA from denaturing and they prevent denatured DNA from accumulating in cells. But it doesn't always just work for heat. It can work for extreme cold as well. But generally think about them as like protecting your organism from a lot of. So they provide higher levels of heat tolerance. There's a lot of studies that have been shown on desert ants and found them to have higher levels of expressed heat shock proteins. There's 6 different families of heat shock proteins, and they each have their own unique evolutionary history, so their own gene tree or protein tree, I guess you could say. And they're not consistently found across all taxa, and they can differ across organism families. So they each have their own unique evolutionary history, and you can create phylogenies of these heat shock protein families and compare them to the organism and see how kind of track them. So as I said, there's been a lot of studies on heat shock proteins on desert adapted taxa, mostly ants and some plants, and they have found that desert adapted taxa have higher amounts of heat shock protein, right? But no one's really looked at flies, and if they have, they've looked at some like cactophilic Drosophila. So these are Drosophila feeding on like rotting cacti in the Southwest, but not really looking at groups like Asteloidea, where, like I said, on the hottest part of the day, we measure the temperature of like the dune sand, and it's like 115, and there's a robber fly just perched right on the sand, or on the little twig a little bit above it, and there's bee flies flying around, super active when everyone is relaxing. So they must have a high amount of heat shock proteins protecting their DNA to be able to function during intense heat. But no one's looked at that. So my research objectives here are my 3 aims are comparative genomics and desert diversification of Asteloidea flies, genome-wide identification, characterization, and evolution of heat shock proteins, and hopefully trying to predict the effects of climate change on asteroid flies. So, someone's laughing. So, the first aim, I'm trying to develop a genomic resource for future studies, so get this library of low coverage genomes, and for that, we're going to use UCEs extracted using bioinformatics and look at divergent times and diversification rate to see how Phylogeia has evolved across their habitats. For genome-wide identity of heat shock proteins, can you identify heat shock proteins using low coverage genomes? What heat shock protein families the asteroid flies have? Do these differ across the various families? Do they differ, let's say, a large family like the psyllids or bombyliids? Do the tropical taxa have fewer heat shock protein or the same amount as one that's endemic to the non-rich sand sea? And are these heat shock proteins under positive selection in desert taxa? That's kind of what we're looking at. And last, hopefully using pastoral biogeography and distribution modeling to see, can we predict, rising temperatures and where asteroid fly is going to move to, and looking at gene evolution of heat shock proteins. Typically a duplication event is shown that protein is under positive selection, and in other groups it's been shown you kind of predict that these species will do better during rising temperatures. So we kind of did some preliminary work with 7 bee flies. We got 7 low coverage genomes. We used 2 different sequencing approaches, so that's why I have NovaSeq and HiSeq. And these are the heat shock proteins that we looked at. And that's just kind of the accession numbers, which don't really matter. Here's kind of the pipeline that I did. And here's some of the results. And this is the one I wanted to show. So of the heat shock proteins using low coverage genomes, we did detect them in a lot of our taxa. Just because they don't have the heat shock proteins, it could just be that they were missed while sequencing them. Quite there yet to figure out, but able to find them in our bombyliid subfamilies, which is kind of exciting. So we know they have heat shock proteins. It'd be weirder if they didn't. But yeah, so for next steps, sequence other asteroid flies, maybe look into longer read sequencing and sequencing the transcriptomes to kind of see if these heat shock proteins are being upregulated. And with that, acknowledgements and questions. Thank you, everyone.
Yes, Ed here. I have a question about the bacteria, the archaeans and things like that, going back to near the beginning of your talk. I'm just wondering, do you predict many unique bacterial species, however you define species, archaeans, protists, and so forth in the psylloid flies? I guess in particular the bee flies.
There might be some unique, so that's kind of getting at where working on microbiomes is hard because a lot of the bacteria are unidentified. So using a shotgun sequencing can really tell you what the bacteria are doing. It's very rare for things to have very unique bacterial species. Sometimes, it's like aphids are the perfect example where they have the most unique bacteria and it's like a perfect system. But for other groups, it's just a lot of different species all doing the same thing. So there might be some unique bacterial species, but there might not as well.
Okay, because I once heard that each kind of insect might have its own bacterial species unique to itself. So you say there's an overlap in the species, but do you think it's possible that there are unique ones with regard to species?
Sorry, can you say the last part again? Can you see that last part again? Sorry.
Oh, I'm sorry, I couldn't hear you. It was a little muffled. So I once heard that there are probably each insect species, however you define a species, has its own unique bacterial species, at least one or so. So do you think that might be possible? You mentioned overlap of bacterial species among the bee flies, but I'm just wondering if that other part might be possible.
Maybe in the future, but right now when you do sequence of microbiome work, a lot of it's unidentified species. Like the bulk of our data would be unidentified species. You'll get it to order or family if you're lucky, but there's so many bacterial sequences out there, and the only way to really describe them as a species is to culture them. You can't culture everything, so it's really hard to figure out what's unique and what's not.
And do you have an idea of the number of archaean species and number of protist species in bee flies?
No, I don't.
Oh, okay, just wondering. just kind of thinking from the biodiversity angle.
Hmm.
Thank you.
Lordotus, do you find them mostly on flowers? Well, they're full of flowers. Have they ever found on the ground where they have composite?
Yeah, like generally on composite flowers, but I have better luck finding them on the ground. You know, they perch on like sandy trail. Like most bumblebees, you have luck finding them like on the sandy trail or like on the wash. So you just find any wash and walk around, and you'll find them perched around flowers.
Do you have any sense of the variation of leg length among your Bombyliidae?
Genus Lordotus or in general?
In general, I just looking across your acyloids, you know, I see like allometries that are surprising to me.
Right.
And some things I can kind of guess like, okay, it's acidity, and so maybe there's something related to prey. But then for things that are primarily flying, some look really stilty, some look really low. Do you have a sense?
Yeah, I feel like most Bombyliid species have like these dainty, fragile little legs. When we're sequencing museum samples in like a big raw like these will rip off a leg and sequence it, but you know, Bombyliid legs are really delicate. So some species have like bigger legs, but most have these really small legs and skinny legs.
What's your like usual collection method for them out in the field? Is it just like sweeping them?
All single net. I mean, you can
Okay.
set them a lace trap, and then you just get a lot of other stuff that you have to sift through. But everything I collected by hand by myself, which is a lot of fun. We get used to looking at stuff.
Going back to the Lordatus rake, I mean, do you think that's partially explained by the relative youth, if you will, of the Great Basin, and that what is desert now not that long ago kind of wasn't, and so these things have radiated very fast.
Yeah, these complexes might just be like incipient species that are just exploded out, which is kind of, I'm hoping to do some like biogeographical analysis and diversification rates to see, you know, how fast these groups are evolving and something like that.
Not a question, but a comment. We should talk more about all of the above because our groups have a lot in common.
I think just because they are pollinators. So have you looked at proboscis link as somewhat of a helpful character?
So that's kind of where the morphometrics comes in. Like the previous authors looked at those and said they were all the same weight. Using an morphometric analysis, you could really, you know, there might be subtle lengths that weren't picked up on a microscope like 40 years ago.
They all have the folding.
Yeah, but they all have the general shape, but they might have different lengths. And the weird thing is like all the male genitalia is almost the same across species, and so is the female genitalia. It's like kind of strange. I mean, within the species groups, there might be some differences, but you know, 40 years ago when these people revised these things, it was like,
You looked at the inner organs,
like beyond the genitalia.
Yeah.
No, I haven't.
Okay.
Is that cognitive well? Look where genitalia were very good. I mean, the groups that I've tried to key off, no, but you know, there might not be keys for a lot of genera that just know. Generally, there's some difference there.
They are the same.
Yeah, they don't do one thing.
One species. So I'm just thinking also about the rake or the big comb, and are there any like very obvious species that are well within these combs?
No, they're all kind of like, you know, they're all just
It
like,
is just a big smear.
it's just like a
Okay.
It's like, here you go. You know, I really when I was waiting for my results, I was so excited because keying them was a challenge and identifying them was a challenge. I was like, I'll finally have the answer. No.
Yeah.
Never going to Kip's office and be like, well, we're still kind of stuck.
how long did it take you to find those?
That's like you think you're in shape and going up with you, and it really like humbles you because every step up is like 4 steps down. And of course, Torson's walking up like no big deal, and it's like, God, I feel so out of shape. It's really windy up there, so on a quiet day it's fun, but on a windy day you just kind of blast in the sand.
How do you collect them so they stay? Because you don't put them in alcohol, do you?
Sometimes I do straight in alcohol. Yeah, you lose the fluff when you put them in alcohol. So that's the unfortunate part.
Just get a hair dry.
Yeah. It all just falls off. So you'll have like a vial, like one of these vials, like a bunch of, I might have a photo somewhere on my phone, all these like golden Lordotus in there. And there's like 5 or 6 from a locality. And by the end of the hike, they're all bald. And then just a big layer of pile and seedy at the top. So when you're extracting, you have to be careful because that will block your extraction tube filter. And so like it forms like a thick mat. So yeah, that was,
Wow.
I guess I'm sure Lepidoptera worked with deal with that scale pretty off. Yeah. So when I sequenced the museum specimens, I got a small paintbrush and I made them go bald, like swipe away all the hairs because there's a lot of grease and stuff that gets caught in that. So I just remove all the seeding.
Now, I'm really curious, has anyone observed one eat clothes from its host?
No, so that's the other way to find it, just to like, they have it for some bee fly species, and the carpenter bee fly here is a very common one, but not for Lordotus. There's like, we think it's this, like this might be this bee, but then that's not it.
What was the carpenter bee fly?
That's like the anthrax that's on around here.
Have you tried to explore their biomes? Because I mean, in other parasitic groups, the viruses play a role in their life history.
No, actually, no one's, that's like, I'm interested in different parasitoids. No one's looked at it from those angles. Like, there's a rich history on Hymenoptera, not so much on Diptera.
Another Hymenoptera kind of thing, cuticular hydrocarbons, do the CD have anything special on them?
No one's looked at that too, so.
So you mentioned that there might be a way to get some of the host DNA, at least if you got it early enough.
Yeah.
Young enough adult. Did you see any evidence of that?
So that was a project that was being done at Berkeley through people at the Gillespie lab. So the person did it with like spiders, and they found like spiders were eating. And so one of my friends did it on parasitoid Hymenoptera from Hawaii. And there's studies that if you sequence the gut content of the adult, you can find remnants of host DNA. So like viruses have fetal DNA. But the issue is when she did that experiment of larger sampling, for some species that are known, like she physically saw them, like she reared them in the lab, pulled them out as soon as they closed, grinded them up and sequenced them and didn't find it at all, no trace of it. So it's kind of like, I think she had a 17% success rate. And so that begs the question of a bee fly, like when it emerges and it has a meal and it passes that meal on, how long does that host DNA last in its gut content, And so it'd be cool to play around if it had a lot of money and time. But when you're doing your dissertation, you don't have those things, so you can't.
What about the prospect of using some sort of sentinel host? If there were some way to put out, I mean, do you have like of your genus? Do you have an idea what any of the hosts associated?
For some groups, I mean, some it's like this genus Phylla, which is a very big genus, has been found on like scarabs and grasshoppers, things like that. So there's some like direction. But it's not for all genders.
So Ed Losowski is in Washington State. Chat. Said, have a brush is supposed to be there if he sees this.
Oh yeah, I'd happily take them. Yeah, alcohol would be best. Yeah.
Said yes. So.
Thanks.
Alcoholic bee flies. I'm curious if isotope analysis might be useful in some way.
Maybe.
Yeah.
It's like a huge open.
Okay.
Want to collaborate? I don't have experience with that, so.
How important is the file for it?
Not for Lordotus. So some groups coloration helps, but luckily Lordotus is like, some species it does.
It falls off the thing.
Yeah. I think there's one species called like Zona that has like a line. There's some that have bands that are distinctive, but most are like silvery, and like males might be golden, females might be silvery, but in some populations they swap. Cool.
Okay, thank you.
Hope you follow.
So we're heading towards the end here of the 1,238th regular meeting. Stop the clock. Talita, did you want to make a quick announcement regarding a possible impromptu ESW talk, or do you want to wait on that?
Yeah, sure.
Do you quickly give a summary?
Yes, for the ESW meeting, one of the people that will be around is Allan. I don't know if I can pronounce her last name well.
I
Well,
can't even remember it, so I'm sorry.
well, she is an ecology entomologist working for the Smithsonian in American Prairie Reserve. She has a great work with climate change. So I was wondering if we could do a next meeting just to have a talk with us because she's been interested in meeting us. She's an entomologist for the Smithsonian that's not here and that doesn't know most of people here.
Yeah, so she'll be in town.
She will be for the ESA meeting. So maybe the day after that, the Thursday 9.
Yeah.
Helen will.
would work. Yes.
Thank you. Yeah, so the idea is we have in the bylaws when the regular meetings are, but there's nothing to stop us from having kind of impromptu meetings when something like this comes along. So would people be interested in something like this?
Oh, that's
Okay.
very interesting.
Okay, good. Then we'll pursue it and figure it out.
If not, I mean, we can always have remote speakers too.
Yes.
It's true.
Yes, but I think she really wants to have like her side to meet us,
Yeah.
not just
Sure, it's
us
nicer in
meeting her. So yeah, I mean that will work if we can make it for the day that she's here. But
Okay. Well, I think this has been a great talk in terms of generating discussion. And we don't all have to leave, but we should. I would like to make a motion to close the meeting.
motion.
Okay, thank you very much. See you next time.